Presentation is the mutagenic properties of artificial sweeteners. Artificial sweeteners are additives that are put into foods to give the sweetness of sugar without having the associated calories. Carcinogenic classifications from Congress and the FDA in the past have brought into question whether artificial sweeteners are safe to consume. For example, FDA members Gene Taylor, Morris Weinberger, and Leo Friedman published a journal describing findings on saccharin-causing tumors, specifically bladder neoplasms, in rats. In the study, 5 out of the 11 total tumor cases were malignant cancerous tumors. In addition, out of the 3 rat groups which were each fed different amounts of saccharin in their diets, the group that consumed the greatest amount of saccharin developed 9 out of the 11 total diagnoses of bladder neoplasms. Studies like these prompted me to create the hypothesis: if E. coli K12 is grown in different medias containing saccharin or sucralose, then the artificial sweeteners would cause changes in the DNA of the bacteria. Research into the mutagenic effects of these sweeteners is important and can lower the number of mutation-related diseases like cystic fibrosis and cancer that occur. In the experiment, the gap of research was that few studies had been conducted on the mutagenic effects of artificial sweeteners like sucralose. The question this experiment answered is, does sucralose or saccharin cause mutations in DNA? The scientific procedures that will be carried out are to first receive the agar slant K12 E. coli, then create the broth mixtures needed to grow the bacteria by mixing sterile deionized water, luria broth, and artificial sweeteners. The broth solutions will then be inoculated and sterilized using an autoclave. The broth solutions will then be placed in a shaking incubator where they will grow for 1 day at 37 degrees Celsius. After the growth period, the bacterial concentration of each sample will be analyzed using a spectrophotometer in order to determine whether enough bacteria is present for a DNA extraction. After connecting a device to the spectrophotometer using Bluetooth, each solution will be graphed and measured for absorbances, or approximate concentration. Then the samples will be transferred to 1.5 ml tubes and centrifugated into pellets of cells. After these procedures, DNA will be extracted from the pellets. The bacteria pellets will be lysed, precipitated, and ethanol washed. Finally, the DNA was run through a gel electrophoresis test where results were analyzed to determine whether visible mutations occurred in the base pairs of the K12 E. coli. After testing, only some wells in the test sample showed visible bands. The bands that were visible had the same size and traveled to the same position in the gel, which shows that no mutations occurred between the positive control group, the 1% saccharin group, and the 1% sucralose group. The absence of bands in the well suggests that the groups of DNA fragments could have either been too small or too large to be visible. In addition, some possible causes may have been that the DNA was stored for extended periods of time, had been degraded, or was incorrectly extracted. It was concluded that there was not enough measurable information to confirm or deny the main hypothesis. An unexpected outcome of the experimentation that occurred during the early phases was when it was determined that the E. coli in the 2% saccharin group was growing at a much slower rate than the rest of the groups. After analyzing the spectrophotometer results, it was discovered that the solutions of the artificial sweeteners had a lower absorbance rate than the positive control, especially both the 1% and 2% saccharin groups. The higher the absorbance rate, the higher the bacterial concentration. The results show a correlation between the increased use of artificial sweeteners and the inhibition of bacterial growth. Some strengths of the experimental design are that there are multiple sweetener types and concentrations which allow for more meaningful and comparable results, due to more factors being tested. In addition, having multiple trials increased the chances of success because I could learn from failed trial groups. Some weaknesses of this design are that the long procedures and time constraints make redoing certain parts of the experiment for better results impossible. Due to a large number of trials being conducted, procedures like the DNA extraction had to be done simultaneously for all samples, which often resulted in certain procedures taking longer than expected. This often resulted in me having to stop the experiment in places where there was no plausible stopping point. Marcus Stafford, a molecular geneticist, helped me throughout the experimentation by teaching me how to keep a sterile environment during the procedures.